Bismillahirrahmanirrahim. Assalamu alaikum. I hope you all are fine and staying safe and happy. I am your science teacher, Miss Farheen, and I welcome you all to your science class. My dear students, as you know that we are on chapter number 2, microorganisms, and today I will deliver its second lecture. So my dear students, let's quickly recap what we did in the previous lecture. Listen to the questions very carefully and then answer them. So my dear students, what are microorganisms? Microorganisms are very tiny living organisms which can only be seen under a microscope. Now, where can you find microorganisms? Microorganisms are found almost everywhere around us. Okay, my dear students, so microorganisms can only be seen under a microscope. They are very tiny living organisms and they are present everywhere around us. And in today's lecture, students will be able to define and describe microorganisms. हम ये देखेंगे कि microorganisms हर जगह हमारे इर्द गिर्द हैं through an experiment. So, my dear students, some pre-reading questions. Can you search for microorganisms around you? Not with your naked eyes, but today I will tell you कि आप कैसे अपने इर्द गिर्द microorganisms को search out कर सकते हैं. So Islamic perspective, I swear by what you see and what you do not see. So microorganisms are present everywhere around us. Even you cannot see them, but they are still everywhere around us. So my dear students, on page number 21, experiment number 1 is given. I request you all to please open it in your science 5 books in chapter number 2. I hope everybody has opened the page. So my dear students, first let's look at the procedure and the material that is required. So to search for microorganisms around you, the material that is required is agar medium, petri dish, cotton swab, and material for agar medium that is required is warm water, yeast which is 6.65 grams, gelatin powder, beaker, and spatula. So my dear students, what is an agar medium? So my dear students, first let's have a look at the material that is required. So my dear students, we need a beaker and we will measure warm water in it 300 ml. Okay, so you can see the measurement here, it is 300 ml. Then we need cotton swabs. I am using these cotton buds and spatula, wooden spatula. I have a glass stirrer here for mixing. Then I have petri dishes. I have gelatin powder and I also have yeast. So my dear students, how we will prepare it? Let's have a look at the preparation first and then we will start preparing the agar medium. So my dear students, for the preparation of agar medium, we will add 300 ml warm water to a beaker, add yeast which is 6.65 grams into the water, mix it well, then add gelatin powder and mix it properly. Allow it to cool. So my dear students, what is the procedure? Pour some agar medium into a clean petri dish. So first let's prepare the agar medium. So how we will prepare the agar medium? So first of all, we will take, this water is warm by the way. This is 300 ml water, मैंने measure कर लिया है इसको. What I will do is I will add 6.65 grams of yeast in it. It is almost all of it. Okay. In total, this contains 11 grams. So almost half or more than half you will pour into this beaker. So if you do not measure it, आपके पास कोई measuring balance नहीं है 6 grams pour करने के लिए, so आप इसकी half quantity को pour कर सकते हैं. So after that you will mix it well with the help of the stirrer. Mix it well. Okay, now once it's mixed, what you will do is you will add gelatin with the help of spatula. अब हम gelatin जो है ये थोड़ा सा ज़्यादा डालेंगे so that this medium will become solid. So water must be warm so that everything will mix in well. So now again mix it well. Once it's mixed, then pour it in a petri dish. So before it becomes solid, यानी कि ये कहीं इसी में solid ना हो जाए, तो मेरी ये petri dish है. I have this. अगर आपके पास ये नहीं है तो you can use any plate, smaller plate. So I will pour it inside it and I will allow it to cool and then it will turn into solid. So क्योंकि यहाँ टाइम मेरे पास ज़्यादा नहीं है, मैंने पहले से अगर मीडियम जो है वो सॉलिड प्रिपेयर किया हुआ है. So you can see it's jelly form, it's solid. So what I will do is now let's have a look कि अब हम प्रोसीजर क्या करेंगे once your agar medium is prepared. So after that, pour some agar medium into a clean petri dish, just like I have put it in. Rub clean cotton swabs at different places around you. अब आपने different जो cotton swabs हैं वो लेने हैं और उनको different places पे लगाना है. Streak the cotton swabs on the agar medium. इसके बाद हम कैसे streaks बनाएंगे I will show you. Leave the streak petri dish for a day. Observe after 24 hours you will see some microorganisms grow on the path along with the streaks. Results and observations, these are microorganisms in your surroundings. Everyday science, keep your surroundings clean. These tiny little organisms can harm you. So you can make a video of your whole activity with the help of your elder and present it in the class. And you can also take a picture and you can paste it here. So my dear students, now let's continue it. So after that, you will take the cotton swabs. I will keep one clean. मैं इसको कहीं भी नहीं लगाऊंगी. I will keep one clean. और बाकी दो को जो है मैं इनको different जगह पे लगाऊंगी. I will put it on the table. इसके अलावा next I will take this one on the walls. Now I will make streaks of it. Here you can see I will make streaks of it on the agar medium. First I will take the one which I put it on the table. और मैं इससे इस पे streaks जो है वो फेर दूंगी इसके ऊपर. Then with the other one which I put it on the table, and then the last clean one. Okay, now let it stay for 24 hours. और उसके बाद what you will observe is आपको मुख्तलिफ जो microorganisms हैं उनकी growth नज़र आएंगी. कैसे नज़र आएंगी? Here you can see the petri dish और इसमें आपको microorganisms की 24 hours बाद growth नज़र आएंगी. These are the streaks जो हमने बनाई थी और after that आपको इसके ऊपर visible जो है वो microorganisms की growth जो है वो नज़र आएगी. And here you can see और ये क्योंकि clean one है तो इसके ऊपर आपको बहुत कम जो है वो microorganisms की growth नज़र आ रही है. So this is an agar medium जो कि हमने gelatin से और yeast से बनाया. And you can see कि different जो microorganisms हैं कैसे वो आपके हर जगह present हैं और कैसे जो gelatin है वो उसके अंदर trap हो गए. And you see them growing after 24 hours. So my dear students, these are the microorganisms which are present all around us. और ये चीज़ हमें ये show करती है कि microorganisms हर जगह हमारे present हैं even though we cannot see them. So my dear students, for homework you can download this worksheet from the description box below this video or you can get it in print form by your teacher. Write down your name and your class, then answer the following questions. What are microorganisms? They are tiny living things which are present all around us and can be seen with a microscope only. How can you see a microorganisms? With the help of a microscope. Microorganisms only cause diseases? Yes or no? Support your answers. No, microorganisms are also beneficial because they are used in many medicines and they are used in many food items. So they are not just harmful, they are also very helpful and they are also used in yogurt which is very healthy. So what did you find by the experiment 1 given in your book, chapter 2? We found out that microorganisms are present all around us. Okay, so we must keep our surroundings clean. So my dear students, for wrap up today we learned that microorganisms are tiny organisms that cannot be seen with naked eye. Microorganisms are beneficial as well as harmful for human beings. So in the next lecture, students will search for microorganisms around them and they will study about different groups of microorganisms. So my dear students, I hope you have enjoyed today's lecture. Keep everybody around you safe. Have a good day. Thank you so much and Allah Hafiz.